Bismillahirrahmanirrahim. First, I'd like to apologize for the delay due to a minor technical issue. It's been fixed. So in this part, I'll continue description of the adaptive immune response. In the first part, in the morning, we talked about the cellular response. Now we talk briefly about the humoral immune response. Again, it's almost impossible to describe the whole humoral response in 25 minutes, so I only included the main concepts of this part of the immune system. So the humoral immune response mainly protects the extracellular spaces. We talked about the T cell component mainly protects the cellular component. However, the humoral response mainly protects the extracellular spaces through the antibodies. The antibodies have three main functions: neutralization, opsonization, and complement activation. We'll talk in a bit of details about each one of these functions, as well as other functions of the antibodies. The structure of the B cell receptor was described earlier, I think, in yesterday's lecture, Dr. Ali lecture. So I removed that part. We'll talk about the function of the B cell. So the classic B cell receptor is an antibody. It's an immunoglobulin. It has binding sites, and it has an Fc receptor. So the B cell receptor has two functions. The first function is the conventional function for any T cell or B cell receptor. It's actually binding an antigen and then initiating a signal pathway to the nucleus, so it actually induces the effector function of the B cell. Now there is another unique function of the B cell receptor. It actually can engulf the pathogen or internalize the pathogen to the intracellular component where the antigen is actually processed and then presented on MHC molecule on the B cell surface. As we mentioned earlier today, the B cell is actually one of the professional antigen-presenting cells. So not only recognizing the antigen, it also helps in producing or presenting antigen on the surface. So as with T cells, B cells actually require two signals or more than one signal to get activated. The first signal is the usual signal that it gets from the B cell receptor. It stimulates activation of the signal transcription. The second signal usually delivered through the T cells, T helper cells, and in particular subset of the T helper cells called the T follicular helper cells that Dr. Abdullah was talking about a few minutes ago. So these two signals stimulate the B cell for further differentiation, proliferation, and production of the antibody isotypes. Now, another unique feature of B cells that is not available in T cells, that B cells can only be stimulated by polysaccharide antigens. Now, we know T cells can only be stimulated by proteins. However, B cells can also be stimulated by polysaccharides. This is a unique feature of B cell receptors, and this can also trigger a signal, and along with the antigen receptor signal, this will be enough to stimulate the B cell to produce its effector function. So the humoral immune response starts by activation of the B cell. So let's talk about how does B cell get activated. The B cell activation, as we said, two signals. The first signal is through the antigen receptor. The second signal is usually delivered through the T helper cell, and this is in the form of the CD40 ligand binding to the CD40. This triggers the signal. There is another help from the T follicular helper cell to the B cells is delivered through the cytokines. There are many cytokines that are actually delivered, helping proliferation and differentiation of the B cells. Among them, IL-21 is the most important signal. So these all signals eventually lead to proliferation of the mature B cells. This is the first mature B cells that get activated. It's called the plasmablast. These eventually differentiate in one of three subtypes of B cells: the germinal center B cells, the plasma cells that produce antibodies, and the memory B cells. Link recognition is an important concept in humoral response. So what do we mean by linked recognition? Now, in order for the T helper to help a B cell to produce antibodies, the T helper should have or must have the same specificity of the B cell. So it can recognize the same antigen that is presented on the B cell. And this is very important for antigen tolerance, for the immune tolerance, because let's say you have a B cell, autoreactive B cells, it produces autoantibodies. But in order to produce autoantibodies, it must have autoreactive T follicular helper cell to stimulate it. So having link recognition actually decreases the chance of having autoimmune diseases. So that's what meant by link recognition. Both follicular T helper cells and B cells should have the same antigen specificity in order to get help. So as we mentioned earlier, naive lymphocytes for specific antigens, let's say an antigen, a B cell or T cell that's specific for Pseudomonas, for example, we only have few lymphocytes that recognize Pseudomonas. Same thing, only few follicular T help that recognize Pseudomonas. So if we leave it for chance for T cells and B cells in the bloodstream to meet, the chance is actually very small. So there is a mechanism that helps to bring these two cells, the follicular T cells and B cells, together in a very narrow area to increase the chance of link recognition and further activation of the B cell. So what is this mechanism? It's again through the chemokine milieu. Similar to what we talked earlier today in the morning about the migration of T cells and B cells, it's the same phenomenon here. So initially, naive T cells express the cytokine receptor CCR7, which directs them to the T cell zone because they're attracted to their ligands, which is CCL21. For B cells, they express the chemokine receptor CXCR5, which is triggered, attracted to its ligand CXCL13, presented in the B zone. These are naive cell distribution, the T cells and B cells. However, upon activation, there is cross-presentation of chemokine receptors between T cells or B cells. So activated T cells start to produce CXCR5, which attract them towards the B cell zone. On the other hand, B cells start to induce CCR7, which attracts them towards the T zone. So the B cell moved this way, and the T cell move this way, and eventually they meet in this small area at the border where the T zone and B zones meet, and this is where linked recognition happens. This is where T cell provides help to the B cell and start to proliferate. Once this happens, the B cells start to proliferate and then eventually start to leave the lymph node and go to the circulation. Plasma cells start to produce antibodies, and some of them go to the bone marrow as long-lived plasma cells, as we'll see in a minute. Now, as we mentioned, in order to have an effect, there must be a tight junction between the T cell and the B cell in order to help each other. And how is this maintained? This is maintained by a series of adhesion molecules. We talked about this in the morning. There are several adhesion molecules. However, they're slightly different than the one between antigen-presenting cell and T cell. Between T cell and B cells, the molecules most important are the SLAM, CD84, and LY108. So these adhesion molecules are similarly expressed between the T follicular helper cell and the B cell, and it maintains direct interaction and stabilized attraction between these two cells, so each one can help each other. So this is an interesting phenomenon. So usually when the T cell binds the T cell helper bind to the B cell, usually the T cell helps the B cell, but also similarly the B cell will actually help the T cell back. So you help me, I help you. Once the T cell activates the B cell stimulates, the B cell get activated, it actually express co-stimulatory molecules, specifically ICOSL, inducible co-stimulator ligand, and this co-stimulatory molecule will help back the T cell to further differentiate and proliferate. احنا عندنا في الحجاز مثل يقول امسك لي واقطع لك. So the T cell help the B cell, and the B cell help the T cell back. And this is one example. This actually mechanism is explained throughout the immune system, and this is just one area where there is feedback help. Okay, so this is what happened at the border. T cells start to B cells start to proliferate. They differentiate into plasmablast. Some of these plasmablasts will move towards the B zone and forming the germinal center. We'll talk about this, and some of them will move to the sinuses and eventually the medullary sinus, and then to circulation. Eventually, some of them will go to the bone marrow. They reside in the bone marrow, and they live there for a long time. And these are actually the sources for most of the antibodies in circulation. So let's talk about the germinal center. As we said, some of the plasmablasts that initially generated will move towards the B cell. And not only the B cells, they actually take with them the follicular T helper cells, so they get continuous stimulation, and this continuous stimulation leads to massive proliferation of B cells within the germinal center. Actually, the germinal center is probably considered the highest dividing cell site in the whole body. Within several hours, probably with every 6 or 8 hours, the size or the number of lymphocytes double in the germinal center. And that's why actually during active infection, we can see that the lymph nodes actually enlarge in patients with infection. Why? Because of the actual enlargement of the germinal centers in the T cell or in the B cell zone. By default, the initially produced or the first plasmablasts that are produced, they usually present IgM by default. So all antibodies first produced in the form of IgM. IgM has low affinity, however, it provides immediate immunity, immediate humoral immunity. So if we have an infection that reaches the blood, the process of having class switching and somatic hypermutation we'll talk about takes time. So you need to have an immediate response trying to contain the infection until you produce the more appropriate isotypes of the antibodies, being IgG or IgA. So IgM is the initial antibody produced. However, we need several types of antibodies. And how does this happen? This happens in the germinal center through two mechanisms: somatic hypermutation, which eventually lead to affinity maturation. We'll talk about this, and class switching, which leads to production or the various isotypes of the immunoglobulins. So what is somatic hypermutation? Now, as B cells proliferate in the germinal center with time, single or point mutation happens in the variable region of the genes, the regions that encode the variable part of the antibody. So these tiny mutations, when they happen, they change slightly the specificity and the affinity of the binding site of the variable region of the antibody. And as time goes, these mutations accumulate, and eventually they actually lead to significant change in the specificity and in the affinity of the end product antibody. Now, interestingly, most of these mutations actually have negative effect. They actually result in either changing the specificity, so the end result antibody actually does not recognize the antigen, or it recognizes it, but it has even lower affinity than the initial affinity. So these cells are actually removed from the cell because they have useless antibodies. So this is a process called negative selection. Less commonly, these mutations can result in production of better antibody who has higher affinity of the same specificity. So these cells are selected for further proliferation. They get more help from the T cell, and this is called positive selection. So positive selection selects the B cells who have better antibody production. They proliferate, whereas B cells with low affinity, they go through apoptosis with a process called negative selection. What about class switching? So in brief, class switching basically means switching the isotype of the antibody with the same antigen specificity. So an important part of this process is the CD40 ligand 40 signaling from the T follicular T cell to the B cell. As I said, B cell by default initially produces IgM. In order for the B cell to switch to other types, it needs help from the T cell, and this help is delivered through the CD40 ligand. CD40 ligands stimulate these enzymes. Several enzymes are involved in this process, and these enzymes start to produce breaks in the DNA. Another series of enzymes repair this break, and eventually they result in formation in a new, different constant region. These mutations all happen in the constant region of the antibody, so eventually the isotype of the antibody change. Note, please, that the variable region is the same. This is the variable region that we started with, and it's the same variable region we ended with. So the specificity is the same. It's actually the constant region that changed, so the isotype changed. So this is just to summarize what's the difference between somatic hypermutation and class switching. Sometimes it's confusing. So this is the initial produce, or the first antibody produced, the IgM antibody. This is somatic hypermutation. Mutations only in the variable region and lead to improved affinity of the antibody. Whereas in class switch, it's the same variable region of the initial protein. However, the constant region has changed, so it was IgM, and this is probably IgG or IgA. There are 5 main isotypes of the immunoglobulins. مجموعة في قولك ماجد, M, A, G, E, D. These are the 5 types, and IgG actually have several subtypes, being IgG1, IgG2, IgG2, 3, and 4. The function of antibody mainly determined by the Fc part of it and by its isotype. So several immunoglobulins have several functions. So what determines what immunoglobulin is produced by class switching? Again, it's all the game of the cytokines. It depends on the cytokines that are produced in the initial inflammatory response, depending on the infection. That type of cytokine leads to production of specific immunoglobulin. So for example, if the main cytokine produced in the site of infection is IL-4, this leads to production of IgG and IgE. If it was mainly interferon gamma, for example, mainly produce IgG and so on. So it's actually the cytokines that leads to selection of certain isotypes of antibody. Okay. We go back to our steps. So proliferation has happened, activation of the B cell, primary follicle has developed, germinal center has developed. Now the B cells start to develop into B cells, plasma cells, and memory cells, as I said. The plasma cells produce antibodies. However, the memory cells, actually I can skip this. Yes, I'm just going to move to the memory cells, yes. The memory cells, on the other hand, they divide very slowly compared to the plasma cell. They have very long half-life. They remain in the circulation for a long time. Some of them actually go to the bone marrow as well, and they reside there. They slowly divide, and they produce no antibody, or maybe very minimal antibody. However, these memory cells are very important for the secondary immune response that was described yesterday. B cells, an important feature of memory B cells, that they already went through somatic hypermutation and class switching. So once they get activated, they define, if they meet the antigen for the second time, they will start immediately producing the high affinity, good quality antibody. They don't have to go through the somatic hypermutation or class switching again. It's already programmed and saved in their gene, so they can produce the high affinity antibody right away, so you can have prompt secondary immune response compared to the first initial immune response. As I said, the function of the antibody depends on the isotype of the antibody. So different antibodies have different functions. I said by default, IgM is the first antibody produced by all B cells. However, IgM only constitutes 10% of the total immunoglobulins in the serum. IgA and IgG are the main predominant antibodies in serum, and IgG actually is the most prominent antibody. I think this is the answer for one of the questions that we started with. Most of this probably because it has the longest half-life among other antibodies. IgE is the smallest concentration of antibody. However, this small amount is actually very biologically active, as we'll see in a minute. We'll talk a bit about each one of these antibodies briefly. As I said, IgM is the first one. It's a large antibody. It's actually pentamer. It has 10 binding sites. Now, each binding site relatively has low affinity compared to other antibodies. However, because it has multiple binding sites, so the affinity, which is the sum of these affinities, compensate for this. So overall avidity of the IgM is actually not too bad. It still can bind to pathogens. Because of its large size, it cannot actually penetrate between tissues. It mainly produces immunity in the bloodstream and in circulation. And because of its pentameric direction or structure, it helps specifically in the activation of complements. There are multiple binding sites for complements, so it's specifically helpful in complement fixation and activation compared to other antibodies. Sure. IgA is a dimer. IgA is, as we all know, it's important in secretions, mostly in intestinal and respiratory tract, also in other secretions such as breast milk, saliva, and tears. IgG present mostly in secretions. So in secretions, we don't have complements, we don't have phagocytic cells. So IgA is actually deficient in these functions. However, it's very efficient in neutralizing antibodies because, again, in the secretions, you have lots of toxins, you have lots of free antigens, so you need neutralizing function, and that's why IgA is efficient in this part. IgG is always monomeric, so it has only 2 binding sites, and that's why it has to have high affinity compared to other antigens. And this high affinity helps it to be one of the best opsonizing antibodies we have. Two words about newborn and humoral immunity. So newborns are very susceptible to infections because their immune system is still developing. So until they produce their own antibodies, they can get passive immunity from their mothers. So IgG is delivered to the baby across the placenta and circulates in the baby's blood. And IgA at the same time is delivered through the breast milk, and it actually infiltrates the infant's gut and gives him further immunity there until it starts to produce its own antibody. I will skip IgE. IgE has main role in allergic reaction, but I will skip it for now. So I'll just briefly summarize the functions of the antibodies. As I said, IgG and IgA very excellent neutralizing antibodies. So what do we mean by neutralization? Toxins have the ability to bind to cells and then engulf inside the cells and dissociate and then perform its toxic effects. Antibodies can actually bind to the toxins, preventing it from binding to the cells, and this is the process of neutralization. You actually prevent the toxin from mediating its effect by blocking its receptors. The same thing happens for bacteria and viruses. Antibodies can actually bind to their receptors and prevent the organism from damaging the tissue. This is the neutralization effect. A second important function is complement fixation, activation of the classical pathway. As we mentioned before, the antibodies can attach to the surface, and then these antibodies have binding sites to the complements, so the complements attach to the antibody, and then this triggers the cascade of the complements that was described by Azam yesterday. And this triggers specifically the alternative pathway of complement system. Another important function, antibody-mediated immune complex. Clearance of the immune complexes. So immune complexes always developed throughout human life. There are lots of immune complexes developed by toxins, debris from dead cells or pathogens, and we have to have a specific mechanism to clear these complexes. Now there are some diseases that were thought, for example, SLE. It was thought that its defective clearance of immune complexes lead to pathogenesis of SLE. So how do antibody help with this? The antibodies attach to the antigen, and then complements attach to this complex, forming antigen-antibody complex. This is recognized by the RBC through their complement receptors, and then the RBCs eventually deliver these complexes to the liver or the spleen, and eventually phagocytosed by phagocytic cells and removed. So antibodies has an important role in clearing immune complexes. Opsonization, this is probably 2 slides more. Also equally important function of the antibodies. Now, phagocytes have receptors for certain pathogens, so they can recognize the pathogen and engulf it, or the other way is through direct phagocytosis. However, some bacteria or pathogens are not recognized by the phagocyte. And in order to overcome this shortage, phagocytic cells have receptors, Fc receptors, to the antibodies. So the antibody will code the pathogen, and this way will help the phagocyte to recognize this bacteria and start engulfing it. So it's a series of binding to the Fc receptors and complement receptors, and eventually it can engulf the bacteria, forming a phagosome, and then lysosomes fuse with the phagosome, forming the phagolysosome. Proteolytic enzymes are secreted, and eventually destruction of the pathogen. So this is opsonization function of the antibodies. Antibody-dependent cell-mediated toxicity. This describes the ability for NK cells to recognize antibodies bound to target cells and helps the NK cell to produce its effector cell by killing the pathogen in exact same way how cytotoxic T cells, or similar way how cytotoxic T cells do that. Antibody-mediated granulocytosis, exocytosis is probably the last function. So some pathogens, for example parasites, are very big, and phagocytes cannot actually engulf the whole pathogen. So antibody can help in that regard. It actually binds to these pathogens, and because of their Fc receptors, they start to accumulate granulocytes around it, and granules are secreted, and this helps killing these large-sized pathogens. This is a table to summarize the distribution of antibodies throughout the tissues that we talked about, which one cross the placenta, which one cross the epithelium. The second table also describes the various functions of the antibodies. As you can see, one single antibody can perform several functions, and one function actually can be performed by several antibodies. So lots of overlap in the immunoglobulin response. And even to make it more confusing, the function of antibody now is discovered it's not only dependent on the antibody itself, but actually dependent on the Fc receptors. So antibodies, IgG, for example, has several receptors: Fc gamma receptor 1, 2, 3, Fc epsilon for IgE, Fc alpha for IgA, and Fc mu for IgM. And these Fc receptors also have several types. And in that